Thank you everyone for joining us this afternoon for today's special topic webinar on the introduction to insecticide resistance in mosquitoes, a case study of resistance in New Orleans. Please, everyone, as we get started, mute your line. If you have any questions, you can ask them in the chat and we'll answer all of them at the end of the webinar. For any additional questions that you may have, you can email them to us at education@nola.gov, and we will be recording this and posting it on our YouTube channel. Today, we have two speakers. We have Erin Cloherty DuVernay and Mark Janowiecki. Erin Cloherty DuVernay holds a PhD in entomology with a concentration in insect toxicology, from Louisiana State University. Erin also holds a Master of Science in Public Health and Tropical Medicine with a concentration in parasitology from Tulane University School of Public Health and Tropical Medicine. Erin has been an entomologist with the City of New Orleans Mosquito, Termite, and Rodent Control Board for 9 years. Her research includes both local and international projects focused on barrier treatments, IPM, insecticide resistance, parasite surveillance, product testing, public perception of pest control, and vector-borne disease control. Mark Janowiecki is a native of Ohio, and he completed his undergraduate degree in entomology at the Ohio State University. Next, Mark joined Dr. Alan Solansky's lab at the University of Arkansas to complete his master's degree in entomology, studying tools to differentiate termite populations and species. Mark went on to complete his PhD with Dr. Edward Vargo at Texas A&M University. His project investigated the physiological and behavioral factors that influence native subterranean termite colony dynamics. In September of 2020, Mark joined the City of New Orleans Mosquito, Termite, and Rodent Control Board, where he is currently running and expanding the molecular laboratory and leading the termite division. And now we'll have Erin start.
Good afternoon, and thank you, Estacia, for the introduction. Okay, so first things first, here's a brief outline of what we're going to talk about today. So why is mosquito control important? What is resistance and how does it occur? The history of insecticide resistance screening methods, resistance levels in Culex fasciatus in NOLA, resistance levels in Aedes aegypti in New Orleans, Louisiana, and what this means for residents. So why is mosquito control important? Adult mosquitoes are a major vector that can transmit disease-causing pathogens in humans. There are more than 1 million cases and over 500,000 deaths from mosquito-borne diseases. Morbidity is also a huge concern of mosquito-borne diseases, leaving many who survive with neurological disorders and unable to work. In 1999, West Nile virus was detected in New York State, and within 2 years, it spread across the US to California and down here in Louisiana. Between 1999 and 2022, there were over 56,000 human cases of West Nile virus. West Nile virus has now become endemic in this country. Last year, there were more than 2,000 human cases of West Nile virus, and over half of them were narrow-invasive. This map shows the number of human cases last year by state, with California having over 250 cases and Colorado having almost 600 cases. In Louisiana, there were more than 46 cases, and that was just in 2023. So mosquitoes are also a nuisance. So if you're getting bitten by one, it's probably one of these three: Culex fasciatus, the southern house mosquito, Aedes aegypti, and Aedes albopictus, and they can all vector pathogens of disease. However, Culex fasciatus is the primary vector of West Nile virus in southeast Louisiana. And that's what I'm going to focus on today. Mark is going to focus on Aedes aegypti later on in the talk. So we're on the cusp of very important and interesting changes happening with mosquitoes right now. St. Louis encephalitis and Eastern equine encephalitis have been reported alongside West Nile virus, and eradicated mosquito-borne diseases are reemerging across the country like malaria, and we're also seeing cases of dengue. So there's a critical need to manage mosquito populations, and there's a number of ways to do that. IPM, Integrated Pest Management, is used by mosquito abatement districts to control mosquitoes. Public education is an integral part of IPM, which is why we're here today. Another aspect of IPM specific to integrated mosquito management is insecticide resistant screening. But before we get into that, I want to talk briefly about surveillance. So surveillance includes monitoring and identification of your pest. Knowing your enemy is one of the first things you need to know to control mosquitoes. Knowledge of mosquito biology is essential for effective control. The mosquito immature life cycle is vastly different from the adult life stage. Mosquitoes are holometabulous, the immatures are aquatic, while the adults are terrestrial. Cloherty fasciatus are opportunistic breeders, meaning they are classified as permanent water breeders, but here in Southeast Louisiana, they're also container breeders, like breeding in waste tires and garbage can lids. They can emerge to adults as quickly as 7 days, which is temperature and resource dependent. Their lifespan is about 2 weeks, and their dispersal is about 1.5 kilometers. Their preferred hosts are birds, then humans, then horses, and dogs and cats. Biting activity is usually dusk, but you'll find them out at dawn as well. And now there are different strategies for control depending on the life stage. What's most obvious is adulticiding. So management of adult vectors, mosquitoes, relies upon effective insecticide use. Only 2 classes of insecticides, organophosphates and pyrethroids, are registered for adult mosquito control in the United States. Both affect the insect's nervous system, but each class has its own mode of action, meaning how an insecticide affects an insect. So pyrethroids are sodium channel modulators, and organophosphates, like malathion and nalid, are acetylcholinesterase inhibitors. And I'm going to delve deeper into that later, okay? But for now, just to put it out there, we wanted to screen Cloherty fasciatus ourselves for susceptibility to bifenthrin, deltamethrin, malathion, and nalid. So both classes of insecticides, pyrethroids and organophosphates, are used by mosquito abatement districts to control mosquito populations on a large scale through ultra-low volume applications. And they are used over the counter by both residents and pest control industry. Repeated use of these insecticides leads to insecticide resistance. And that leads me on to our next section. What is insecticide resistance? So I'm going to give you a definition, right? Insecticide resistance can be defined as the developed ability in a strain of insects to tolerate doses of toxicants, which would prove lethal to the majority of individuals in a normal population of the same species. So here we have a naive population with some mutations, right, in this slide. They're sprayed repeatedly and offspring develop with mutations, and so on until we have a resistant population. So insects have the ability to resist toxic effects of chemicals. Just in nature, they're exposed to different environmental factors that produce these toxins. Minor genetic mutations can occur that make insects resistant to new chemicals, and mutations arise at random. So let's talk about the history of insecticide resistance, all right? So my bachelor's is actually not in science, it's in history. So I find this part very fascinating. In the early 1900s, the first documented case of insecticide resistance was to sulfur lime in scale insects. And then you had arsenic resistance in gypsy moth caterpillars. And up until the 30s and 40s, there wasn't much documentation of resistance when they started using chemicals to control pests during World War II. DDT and organophosphates were developed from nerve toxins. DDT was used to fumigate for fleas and lice and mosquitoes and to prevent diseases like typhus and malaria. In the 1950s, insecticides became less effective DDT resistance was first documented in houseflies. There was a resurgence of pests after treatments, and the Metropolitan Mosquito Control was formed, along with the Louisiana Mosquito Control Association in Louisiana that used DDT to control mosquitoes. In the 1960s, Rachel Carson published Silent Spring. So this novel had a huge impact, right? She published information about adverse side effects on wildlife, water quality, and human health. The DDT biomagnification in foods and milk. This is when the environmental movement really starts to begin in the US and abroad. Pyrethroid insecticides were still under development, and mosquito abatement districts start to be formed across Louisiana. In the 70s, government public policy changes. The Environmental Protection Agency was created. The USDA created nationwide IPM programs. DDT is finally banned in the USA 10 years after Rachel Carson's book was published, and resistance is being reported to pyrethroids. Moving along to the 1980s, we have continued use of pyrethroids and organophosphates. Pyrethroid resistance is growing. We've got GMO crops with target pesticide resistance. Emphasis on cultural practices are reinstated for agriculture, and Food Quality Protection Act was passed. Now, in the 21st century, resistance is still widespread. DDT is still used in Africa and developing countries. Pyrethroids and organophosphates are still used to control mosquitoes in the US. We have emerging and infectious diseases are worldwide, and we have over 500 species of insects that are resistant to insecticides. So it is important to screen for insecticide resistance. Ideally, you want to make sure the insecticides you want to use work before you use them. However, sometimes this is not a possibility, but you still need to screen so you can plan more effective control strategies. So there are various insecticide-resistant screening methods available. We looked at the CDC bottle bioassay kit and the topical application assay. So the CDC bottle bioassay kit comes with insecticides and everything you need except mosquitoes and acetone. So first you mix and coat your bottles with the insecticide, you transfer your mosquitoes and record mortality at the diagnostic time. Mortality is determined by the inability of the mosquito to right itself when it cannot stand or they have erratic behavior. The CDC kit uses a diagnostic time and a diagnostic concentration to determine the proportion of a population exhibiting resistance. These are the reference points against which all field mosquito results are compared. Diagnostic time is the expected time for the insecticide to kill nearly 100% of susceptible mosquitoes. The diagnostic concentration is a concentration of insecticide that kills nearly 100% of susceptible mosquitoes by the diagnostic time. So the CDC has optimized the kit with diagnostic concentrations and times for several insecticides and species of mosquito. Ideally, it kills 100% of the population within that time. We wanted to test deltamethrin, malathion, nalid, and bifenthrin, and we wanted to test Arculus quinquefasciatus population. So if you look at the deltamethrin under the insecticide column and follow it across, each bottle will be coated with 0.75 micrograms of the diagnostic concentration, which should kill 100% of the susceptible Culus quinquefasciatus in that bottle in 60 minutes. If a field population tested with the same diagnostic concentration survives past that 60-minute diagnostic time, they are considered resistant. Now, if there's a chemical that's not listed, like bifenthrin, the CDC will send you the insecticide concentration and you can figure it out the diagnostic time yourself. However, you will need a susceptible strain to do that. Now, for the topical assays, you need to establish discriminating doses yourself, as the CDC bioassay did, using a susceptible population of mosquitoes. Now, we wanted to estimate the LD95 of the susceptible strain and then apply it to the field population. So just a few terms is you got lethal dose, which is a fixed amount of an insecticide that kills a percentage of insects. You have the LD50, which is the lethal dose that kills 50% of a test population. And you have the discriminating dose, the dose that discriminates between susceptible and resistant in a test population. Any that survived a discriminating dose is resistant. Now, once you have established your LD50, you can estimate your discriminating dose, the LD95, using probit analysis. Log dosage probit mortality regression lines can be calculated using computer software programs or estimated using log probit paper. And to do that, we had to run a series of doses to run a line and extrapolate values corresponding to an LD95. And we did this for each of the insecticides we used in our field populations. So to set up for a topical application assay, you first need to prepare your own chemicals, which requires, again, a susceptible reference strain of insect to be tested before and alongside your field population. You do this by making serial dilutions with your technical grade chemical of choice and testing a susceptible strain prior to testing your field population. Then you anestheticize your field mosquitoes and apply the LD95. Mortality is read at 24 hours, and you want to determine it, or rather we determined it in the same manner of the CDC kit. So here we've converted our data for the topicals into log dose and probit mortality to calculate our discriminating doses. We applied our LD50 and estimated discriminating doses for a susceptible strain. Our calculated LD95 killed 95% of our susceptible strain. So if you look at this side, you have log dose in the x-axis, probit mortality in the y-axis. Bifenthrin is in orange and deltamethrin is in blue. The dotted lines denote our LD95s. Here we have the same for organophosphates, malathion, and nalid. Again, the LD95 is in the dotted lines. Now, once you've determined if you have your insecticide resistance in a population, you want to figure out why you have resistance. So, how does insecticide resistance occur? Okay, there are three main mechanisms of resistance. There's reduced penetration, metabolic resistance, and altered target site. Metabolic resistant insects develop enzymes that detoxify pesticides more rapidly, preventing the active ingredient from reaching its toxicological target. Altered target site resistance, the protein target that the insecticide is designed to attack, is altered by a mutation, and the reaction doesn't end in lethality. We looked at metabolic and altered target site resistance in half of the sites that we screened for susceptibility. We measured alpha and beta esterases and gluta-S transferase. So esterases in insects break down esters, including the two classes of adulticides, pyrethroids and organophosphates. At the top of this slide is the chemical structure for bifenthrin, and like other pyrethroids, it's an ester. And at the bottom of the slide is the alpha-naphthyl acetate. And you can see the arrow pointing to the ester. Now, if the ester were on the second carbon, where you see the beta sign, it would be a beta-naphthyl acetate. So esterases attack the ester and they hydrolyze both alpha and beta naphthyl esters, producing the color we see in the 96 well plate. The more esterase activity, the more colored product you see. We measured optical density using a spectrophotometer to determine enzyme activity. We used a similar approach to measure glutathione S transferase, which have been shown previously to be important for pyrethroid and organophosphate resistance. So pyrethroids also target the nervous system. They bind to a protein called the voltage-gated sodium channel. Normally, the sodium channel opens, causing stimulation of the nerve, and closes to end the nerve signal. Pyrethroids bind to a receptor on the sodium channel and prevent it from closing. This causes prolonged action potentials, restimulation of the nerve, repetitive firing, hyperexcitation, and eventually death. So insects have modified the target site. So an insecticide cannot bind to the receptor. The sodium channel, which you see here, right, pictured, is a huge membrane of proteins with a lot of potential mutations. The actual mutation that we looked at was for a leucine to phenylalanine amino acid substitution at this site circled here for the L1014F mutation, which is also the most common mutation in insects. And we used a PCR assay to detect the mutation. So this is also called the knockdown resistance mutation, or KDR, and it only affects pyrethroids. There are several and different mutations for different species of mosquitoes. So the L1014F is more commonly found in Culex, whereas Mark will discuss later the KDR mutations that are more commonly found in Aedes. Okay, let's talk about our case study in New Orleans, Louisiana. Okay, now New Orleans lies east of Baton Rouge and southeast of Lake Pontchartrain. The map shows the city of New Orleans divided up into their mosquito surveillance control zones. Site selection was based on these zones. Now, Culex egg grafts were collected from 12 of these zones from July through October in 2021. Egg grafts were hatched out in separate containers, and larvae were identified to species and then pooled and reared together for each site. Adult mosquitoes that emerged from those egg grafts were screened for susceptibility, and again, to bifenthrin, deltamethrin, malathion, and nalid. Okay, so we have the mean resistance frequency side by side for all 12 sites for both assays for each insecticide. So as you can see, there's high frequencies of resistance detected at all sites. Both assays do detect resistance in the same populations of mosquitoes. However, you can see there are differences in frequency of resistance between the two tests. Look at the mean of the bottle versus the topicals. There are vast differences, right? Pyrethroids are much lower in topicals than bottles. Organophosphates in bottles are much lower than topicals. So again, these are the means of all 12 sites, but they don't really capture the diversity or the differences between bottles and topicals. So here are all the individual data points from all 12 sites. So if you look at malathion, right, that's highlighted in yellow, there's diversity across all sites in the bottles and the topicals, and some are more susceptible than others. And at individual sites, there are huge differences between bottles and topicals. So for instance, at site 5 and 6, right, that are highlighted, you go across to malathion and you have 19% resistance in bottles and 100% resistance in topicals. It's the same for site 6. Bottles are 37% and topicals are 100. But then if you look at site 9, bottles and topicals are similar at 36 and 33%. So yes, both assays report resistance, but they are not the same. Now this is less problematic with pyrethroids, but still, if you look, like site 3, bifenthrin highlighted in blue, you have bottles and topicals 91 and 90, deltamethrin, bottles and topicals 93 and 100. So when we regress all the means from all the 12 sites for both assays, they are poorly correlated. All right, so next we looked at mechanisms of resistance, why we have resistance, and we looked at metabolic resistance first. So if you look at this table, you can see the variability among sites for both alpha and beta esterases. we have the mean of enzyme activities measured in our susceptible strain and field populations at 6 sites. Our field population is much higher than our susceptible strain. Alpha esterase in field populations was 67-fold higher than the susceptible strain. Beta esterase in field populations was 24-fold higher than the susceptible strain. And GST was almost 3-fold higher than the susceptible strain. So enzyme activity was variable across all 6 sites. And for esterase, that variability was correlated with resistance frequencies, as seen in the top graph, right? So the strongest correlation between esterases and resistance frequencies were with deltamethrin resistance frequencies using topical assays. Correlations were not as strong between bifenthrin resistance and esterase activities, and correlations were poor between resistance frequencies of organophosphates and esterases. And interestingly, alpha and beta esterase activities were correlated in insects from these collections, meaning that if you had high alpha esterase, you had high beta esterase enzyme activity. So here we have a comparison of the frequencies of the L1014F allele for the susceptible strain and field population, right? The KDR. As you can see, frequency of the L1014F mutation was high and variable across all sites. However, frequencies were not correlated with insecticide frequencies of resistance. About 70% of all the populations have the resistant allele, but only the RR are resistant, 52%, where the RS heterozygous are just carriers. So again, KDR frequencies were not correlated with pyrethroid resistance frequency in either screening method used, which can translate to just because you have mutations does not mean you have resistance. So in summary, both assays can detect insecticide resistance. However, the correlations between results from the 2 tests were poor. For abatement districts, the CDC bottle bioassay kit is a lot more user-friendly and more applicable than the topicals. So our next question was, what mechanisms are associated with the resistance we found? So results suggest that activities of alpha and beta esterase, and to a lesser extent GST, are associated with pyrethroid resistance in Culex fasciatus. The L1014F mutation frequency, or KDR, was high in these populations, suggesting that target site resistance was present, but it was not correlated with resistance frequencies. And the enzyme activities measured here do not appear to be associated with organophosphate resistance. So with that, I would like to acknowledge the New Orleans Mosquito, Termite, and Rodent Control Board staff, Louisiana State University Department of Entomology and the Ag Center, and the East Baton Rouge Mosquito and Rodent Abatement District. And from there, I'm going to leave it to Mark to carry on.
Awesome, thank you. If you want to stop sharing, I can start sharing.
Okay. I think you're good.
Perfect. Okay. Can you see my screen now? Okay, awesome. So yeah, I'm going to take over and kind of switch gears from Culex into 80s. And so this was a big project that came about when Zika was around, or was first around, and was trying to really determine what's going on in our Aedes aegypti populations in terms of pyrethroid resistance. And so the goal here was to look across the city, again, those spray zones that Erin mentioned earlier. We have all of these zones across the city to characterize resistance across all of these zones to see if it's the same everywhere, if there's pockets of resistance. So we did bottle bioassays for organophosphates and pyrethroids, as well as the knockdown resistance KDR screening with molecular methods for just pyrethroids only. And so we had that same kind of question that Erin was just asking of, do different methods of surveillance correlate to each other? Because these methods have different levels of effort and work to put into them as far as rearing mosquitoes versus just passively collecting extra ones out of our surveillance that happens weekly. And we just wanted to know if these were giving the same answer or if we needed to use one or the other. So our methods, as Erin discussed, we looked at organophosphates and pyrethroids. We had 2 organophosphates and 5 different pyrethroids. We ran the bottle bioassays to see the mortality over time when exposed directly to these chemicals. And then we also did the KDR analysis with molecular methods, looking at the susceptible and resistant haplotypes with melt curve analysis to see if how the mutations look, and they should melt, the DNA strand melts at different temperatures, which we can determine as you see in the graph over there. And we looked at 2 different KDR mutations for this study. They're just named KDR3 and KDR5 for simplicity, but just in the order that they were discovered, and kind of the relevance here is we have a lot of KDR3 and KDR5 mutations in the US. In other areas, there's other mutations as well that are occurring. So I'm going to jump into the results because Erin did a good job of showing a lot of those methods of how to do these bottle bioassays and how the KDR works. And so just in case you're not familiar with how this spray map is, this is the city of New Orleans, and I'm going to do a quick New Orleans tour just to kind of orient to where all these different zones are. So obviously the Mississippi River is flowing along the city, the Crescent City there. You can see the Mississippi River flows along. Let me see if I can get the pointer here. Yeah, so the Mississippi River runs along the south side here and goes along, cutting off the West Bank area. So that is the river. The French Quarter is this zone right here along the river. City Park is located a little further up north in the city. Audubon Park is down by the river in the southwest. Our building, which we're located here in Mosquito Termite Rodent Control Board, is up close to the lake. And then, as I mentioned, this is Lake Pontchartrain, which is the northern border of our city. So when I put the frequency of survival of individuals in the bottle bioassays, again, this is, even though some are red and some are green, the difference is not very large. This goes from 0 would be the darkest green individuals surviving the bottle bioassay, so all of them died from the pyrethroids, up to about, I think the highest one there is between 6 and 6.75% of individuals survive. So even though it looks red, I just had the dramatic colors so you could see the differences, but this isn't really showing dramatic differences occurring. Although there is a small hotspot over here. And so this hotspot is in zones I1, I2, and H1, is a little more individuals surviving the bottles, indicating that there's some resistance going on a little bit more in this area, but nothing of concern with these results. There's also some of these yellow ones kind of intermediate in L1, O1, and P2. When we look at the percent of individuals surviving the bottle bioassays for the organophosphates, the map is a lot more green, which is what we want to see. So there's a lot less individuals surviving, or a lot less potential resistance occurring out there in these areas, although we still have this one zone I1 where, up to over about 9% were surviving the bottles in this zone. So we put them side by side, as it's rather obvious there's more resistance in pyrethroids than organophosphates. So our organophosphate chemicals would be expected to be killing more mosquitoes and working more efficiently in the field compared to our pyrethroids. And there's this kind of zone that something weird is going on in I1, where there might be some resistance developing or something occurring there. So then we took all of these individuals that were running the bottle bioassay and did them specifically, individually, pairing them the data from the bottle bioassay to the KDR screening. And so with this, the table here shows the 2 different KDR mutations. So on the left side is KDR3. The VV would indicate that they're susceptible. The VI would be a heterozygote, and then the II would be the resistant haplotype for each of these. So across the top is KDR5, with FF being susceptible and CC being resistant. So individuals in this kind of top left corner, which is about 95% of these individuals, had neither mutation, whereas individuals down here with about 0.8% had both of these 2 mutations. So looking at this, And this was with a very, very large number of samples. So this was 25,543 individual mosquitoes that were run through first the bottles and then through the KDR. There was a portion of the samples that we kept for other parts of the study, and some of the KDR screening didn't work successfully, so we only had about 98%, but that's still more than 25,000 individual mosquitoes that we looked at to get these numbers, so we're very confident in these percentages. And the vast majority are susceptible to pyrethroids. So for the most part, this looked really good. So we saw this and we said, you know, our chemicals should still be working on a very vast majority of the samples that are out there. We should be in good shape. Putting these KDR results on the map. So this is showing the numbers. So now instead of having survival in the bottles, this is the average number of KDR alleles per individual for both loci. So if they're totally susceptible, they would have a value of 0 because they carry 0 of the potentially 4 alleles they could be carrying. If they have, you know, just a mutation in one of the KDRs and not the other, that would be the 2 because it has 2 alleles from one, 0 from the second. And an example of being at a range of 4 would be something where it has the mutation in both KDR3 and KDR5. So overall, these are very low on average, as you would expect, since 95% of the samples we looked at didn't have any. Again, we have this hotspot still in I1. This time it's slightly different, but still kind of around this area with K2 and L1. But of note, interestingly, 8 of these zones have no KDR alleles present. So this dark green means no individual in that collection had either of these mutations at all. So those completely, you know, purely non-mutated populations in those areas. Most zones have pretty low levels, and then again, kind of the same story. We have that small pocket of resistance in this mid-city area. So the next question, once we knew, you know, we're in pretty good shape, but we have mostly susceptible mosquitoes out there, was the question of how well do these correlate to each other. So just by looking at the map here, you can see they do overlap in that hot spot. So both of them, you know, identified this area as having, you know, something going on. But there are small differences, as you can see with the map. There's differences in some of these areas. So what I did is ran a correlation of these different samples. And so on the x-axis, we have the number of alleles going from 0. Again, this would be totally without either of the mutations to 4 with both mutations. There's different ways they can get to have 2, 3, 1, 2, or 3 alleles. And so those different haplotypes are different dots on the graph here. And then on the other side, we have the log scaled percent survival. So that's why it's so low in this case. But this log scaled percent survival indicates how many survived out of the bottles. And we have a very strong correlation here, or a relatively strong correlation, where individuals with more KDR alleles were more likely to survive the bottle bioassays. So the individuals that had both mutations were much higher survival compared to ones that had none. So we were happy with this and finally completed this data set. It was a lot of work to get all 26,000 of these samples through and a lot of time. It took us, you know, until last summer to wrap up all of this molecular analysis with lots of people helping. And as we were wrapping up, I was curious what it looked like now. So in this last year, in 2023, in the season of mosquito season, I had our surveillance team pull out any of these Aedes aegypti mosquitoes that they had extra in their trapping so we could try to see what it looked like now. And so with this, I ran about 180 samples, so not quite the 26,000 of 2017, 2018 of these samples that we ran, but a good number to get a pretty good idea. And of these, this table now looks very different. So what used to be 94.5% of them had neither mutation. Now we're looking at, you know, 3% of them are in that category. And we have a lot more in this area over here. What used to be, you know, 0.8% of individuals had both. Now almost half of our mosquitoes are carrying both. So in the course of this, you know, 5-6 year period, we've completely changed in the city, and it's completely flip-flopped. So putting that in perspective on the map here, we have the map of 2017, 2018, and then now again there were some zones that we didn't get collections from, so that's in the gray there. But where we did, it's completely changed. So now it's a massive red spot that we're looking at, and that's using the same scaling for those categories there. If we scale it a little bit differently and up it to now, instead of just going from 0 to 0.8 average KDR alleles per individual, now we're going from 0 to 3.6. Now you can see a little bit of differentiation between these groups, and interestingly, our hotspot that used to be is one of the cooler spots relatively, whereas kind of this French Quarter area over here and surrounding area now is a slightly hotter spot. But again, it's pretty ubiquitous across what we've looked at in the city, and we wouldn't expect it to not kind of fill in some of these gaps. And so this summer, we're planning to do quite a bit more sampling and really make this a more robust assessment. But I think from this preliminary data, there's no way that it's not going to be somewhat different from 2017 to 2018 or to now. So overall from this, initially in 2017, 2018, we had very low levels of resistance, about 0.8 of individuals, whereas now we have about half of our individuals. So this has completely changed. We're in line with different areas and around the world. They've checked for these mutations. Some areas like Delhi and India had very low, similar to what we used to be. Florida has been tested, some populations there, and found to be what we are about now. And then some areas in Houston are even worse, in Harris County, Texas. And so resistance obviously can and is changing over time. So it really matters what we do, product stewardship and what we do and what the homeowner does as far as what's going on to change these populations. In Houston, for example, as I showed, it was 77% in a paper that came out in 2021, looking at, I think, around 2019, 2020. But a paper that came out looking at this in samples from 1999 found no alleles present. So they had no mutations at that time. And in the course of almost 20 years, it's gone up to 77. So I think we're on our way to kind of have that same pattern occur as well here. And laboratory resistant selection trials, where they just take insects, expose them to high levels of insecticide, and then rear their offspring to try to get as resistant of a population as possible. In these, very fixed conditions, they were able to drive these to fixation after just 5 generations. So the mosquito generation time is very short. This is very feasible to really make a change in our populations that are out there. So overall, in our 80s population and with this big data set, our 2 assays were about comparable. There's definitely slight differences in which ones were, the most or the least resistant, but they're showing about the same thing. They're showing comparable levels. And our 80s populations in New Orleans, unfortunately, are developing resistance to pyrethroids. So this is a change from the initial data set in 2016, 2017, where we can see this happening in real time. And again, as I mentioned, this was done with a lot of support. This is acknowledging a lot of the funding and the assistance in getting the KDR running, as well as the staff that was able to run all of these bottle bioassays. I forgot to put on here, but we've also had many staff members, some of which I think are on this call. We have Grant here, who has helped out with a lot of these KDR analysis in the lab for the lab portion of this as well. So to run 26,000 samples is not something that happens quickly and without help. So this has been a big group effort. But what does this mean? So what does this mean for a homeowner? What does this mean for us? We are certainly considering what products we're putting out and what mosquitoes we're targeting, and being aware of if we need to consider that these products aren't going to be working anymore. But what about these backyard sprays? So the backyard barrier treatments are usually using pyrethroids. And we know the West Nile vector, from what Erin showed, is resistant to these pyrethroids, and we're developing resistance to these pyrethroids in our 80s populations. So what does this mean? Obviously, this is showing that this repeated use of the same insecticide can lead to resistance. Sure, if you're not targeting these public health pests, if you're after other species, we don't have the data, but certainly what would be expected is if you're repeatedly using these, if you schedule your backyard spray every month or several times in the summer, this repeated use of the same insecticide is going to lead to resistance. So it might be something to consider, looking into what you're targeting and how well these insecticides are actually going to work, because you're paying for this and there's a cost, and you're impacting the population around you and taking these tools away from us if we're in need of these tools to still be working down the road for mosquito control. But I think for the homeowner, what I want to really just reiterate here at the end, that I think we've seen in other webinars, and hopefully you've seen through our social media and all sorts of things, the thing that you can make the most impact with around your own house and neighborhood is source reduction. So we need to think about where the mosquitoes are breeding and try to eliminate these sources of breeding to really cut down on the number of mosquitoes around. It's not just something that we can spray and hope they disappear. This is something that's tried and true. They can't be resistant to you dumping their water out. So let's really focus on these. So some of these are, checking your property for standing water. These mosquitoes, some of them can breed in very, very small amounts of water, enough just in a single bottle cap or different areas. You can regularly turn over containers and remove clutter. So remove things like tires, get them picked up. We can, if you do a bulk trash pickup, the city can come and pick these up. Check storm drains and gutters and flush them if needed. So be aware of where water is pooling and where mosquitoes might be breeding. Dispensive items that hold water. So these can be tires, as I mentioned, plant trays. Look under the tray under your potted plants in the backyard and see, is there buckets that have just been tossed outside and left? Tarps can often have folds that hold water. Ice chests can be left out. Kids' toys, all sorts of things can be left out. Once a week is approximately sufficient. Usually the minimum time from egg laying to adult is 6 to 7 days in peak season. So if you just go around once a week when you take your trash can out to the street, just go around your property and try to do this and educate your neighbors, because what your neighbors do really is going to impact you. Some of these mosquitoes have very short flight range of just, within a couple blocks. Some of them fly further, but some of these things, can really be impacted by what's going on right next to you. So talk to your neighbors, talk to, different people, and try to educate them on these same ways of cutting down mosquito breeding sites. And if you need to, we can conduct yard inspections. If you're not sure where the mosquitoes are coming from, call in 311 and put in a request. We'd be happy to send someone out and see what's going on and try to help get to the bottom of why there's so many mosquitoes. But really, all of this, the source reduction, as I mentioned earlier, is something that they can't become resistant to. If there's no water to breed in, they're not going to adapt to breed without water. So this is a method that is always going to be in our arsenal. It just takes more time and effort. It isn't something you can hire a pest control company to come in and spray in an hour and take care of this issue. It's a longer term battle. But these are all examples of some of those water holding containers. So with that, we'll wrap this up, and I'd be happy to take any questions if we have them in the chat. Again, follow us on our social media. We put out a lot of tip and toss, different examples, and all sorts of future webinars and things that will be coming up for training and education. But yeah, we can see if there's any questions.
Yes, Mark. So the first question, Mark or Erin, can you speculate on what is driving the change over the last 8 years compared to the previous 8 to 12 years? Pyrethroids have been around for longer than 8 years. Increase in mosquito-specific pyrethroid treatments by PMPs?
Yeah, that's a great question, and one that I would love to know the answer to. Yeah, what's gone on there? Yeah, they have certainly been around for a longer amount of time. I think seeing this change now, we're going to be, keeping our finger on the pulse a lot closer to see if, are we going in a direction, or does it fluctuate over time? Storms bring in newer populations, or different things could occur. But I don't know, and that's the problem. I don't know if Erin, if you want to weigh in.
Sure. Yeah. So I think for for either species, it's interesting with the 80s, right? Because that we're seeing that 2016 to now. I mean, like, that's really cool. Actually, this research wise, operationally, it's not. But yeah, I mean, like, if we haven't tested the Culex in over a decade, right, for resistance. But you look at like historical data, they've had pyrethroid resistance, for 10, 20, going back 30 years, right, in these mosquitoes in the greater New Orleans area. And so abatement districts don't spray normal. They did used to spray resmethrin, some deltamethrin, right? But like New Orleans doesn't use pyrethroids as much, except for, we spray on data-based activity, right? So if we have a high level of Culex and we have West Nile positive mosquitoes, then we're going to apply stuff. But so why we have this resistance, I mean, we really don't know. We've done some barrier treatment trials to see if we can to show resistance in the field, to see if, you know, backyard spraying is causing it. And or if homeowners are buying stuff off the shelf and that's adding to it. But we really don't actually know with the pyrethroids. So again, like it's a $1 million question is, you know, why is this long term? And then once you have the resistance, it is with pyrethroids, it is very, very hard to reduce that. So I mean, with the Culex fasciatus, you know, our resistance levels being, you know, 90, 96%, it's going to be rough, right? But if we can, you know, find other sources and methods to control that, then, I mean, that's going to be our best bet. So, yeah, can't really answer your question there, you know, but I can speculate. There we go. It's just, you know, long-term usage, you know, and then, you know, and then it's a question also is like, you know, is it in the, you know, is it in runoff somewhere, right? Or, you know, because it's still getting used, right? People are going to use it, whether it's using for mosquito control or not. Pyrethroids are used for other insects, right? So it's there, it's in the environment, and it's being used in the environment repeatedly. So, yeah, I can speculate, but I can't give you a definitive answer.
Okay, Mark, this question is for you. What do you think could account for the low to no KDR present in some of the study areas? Any activities that could point to this? Are you rotating the classes of insecticides?
Yeah, so the low to no KDR was only in the data from 2016 and 2017. So in the most current data, there were pretty even levels, and it was definitely everywhere. So I mean, I would think that at that point it was a pretty naive population that just hadn't had those mutations in there. I think it's different pockets, and I don't know. It would be interesting to, you know, if we had the records of how many backyards were sprayed and when, and see, you know, if different neighborhoods sprayed more, or, you know, if we could track that. I think that would lead to that, kind of as Erin was talking about. But we don't have that whole data set, so we don't know exactly what's causing, you know, this to be more or less mutations. And as far as, yeah, rotating classes of insecticides, yeah, we use organophosphates and pyrethroids, but the issue is there's not a lot in our tool belt to be rotating much further. I don't know. It's we need some more different active ingredients to use on mosquitoes to really do a better rotation. But organophosphates are the one that seems to work, and we tend to use it more. But we do rotate when able, I would say.
So I just want to add on to that too. So what's interesting, especially with the 80s, right? Because Mark just tested on this, like, okay, 2023, is that we haven't been using a lot of chemical, either class, since 2021, since we saw that our Culex were resistant to pyrethroids and organophosphates, right? So the only thing that we have been using, like, you know, when we go to spray the cranks, we're going to be spraying the 80s are going to be exposed, right? Like, you know, so it's just very interesting that we are seeing resistance because the last 2 years we haven't really been spraying, we've been larviciding, right? So that to me is like the most interesting thing. It's like, okay, where is this resistance coming from? Why are we getting these mutations? Right. So it's, yeah, it goes hand in hand, all those questions. But I think that's the really interesting part. It's like, yeah, okay, we do rotate, you know, or we have in the past, but we haven't been using anything in the last couple of years, you know, unless we had a West Nile positive, you know, in Orleans Parish, we don't spray on a weekly basis just to spray. Right. So we need a causation for that. So yeah, I think that's that's the really interesting, I think, aspect of it.
Okay, thank you both for your answers. I don't see any more questions in the chat, but just a reminder, if you have any questions that you think of, you can email them to us at education@nola.gov. I want to take a moment to go over a few upcoming webinars we have going on. So, first we have Mosquito Academy coming up April 23rd through 25th. That'll be a 3-day intensive course with 1 day of all lab work with ID and mosquitoes. So if you want information on that, you can email us at education@nola.gov. We also have the last part of our rodent webinar series. So that will be Thursday, February 29th, and we'll be talking about the importance of rodent management. There are CEUs available for sanitarians for that webinar. And then after the rodent control series, we'll be starting the mosquito control series in March. So that's also coming up. So if you're interested in attending any of those webinars, please email us and let us know. But once again, thank you everyone for coming out. Have a great day.
Thank you.
Thank you.